Alright, so this is a huge DNA molecule, right? If I were to count every 3, I could call that a triplet. Every 3 letters of messenger RNA, I'm going to call a codon. That you do need to know for the quiz today. Alright, and then every 3 letters of tRNA is called a what?
Anticodon.
And what does tRNA carry?
Amino acids.
Amino acids. Okay, so we've got here, can you come hold this for this one? Alright, so here's our DNA. Alright, hold that and I'll take it from you a second. Here's our DNA. Here's the RNA. 2 hands. And then this is the, when I said RNA, did I leave something out?
Messenger RNA.
Messenger RNA. A lot of biologists do that because once the tRNA is made, it goes to the cytoplasm, you never really see it in the nucleus anymore. But messenger RNA is constantly leaving the nucleus, going to the ribosome, where it's going to do translation, and then it's going to go back to the nucleus, get cut up in small pieces, be recycled. The ultimate renewable resource is when another DNA is going to code for it and make it. So these are the players. There's tRNA, and then this is amino acid. Actually, if you look close, you can see the name of it. It's called serine. Right? That's the only one. It's one of the 20. Okay. Let's see this. Is DNA, what does it mean when we say a coiled molecule? Or a twisted ladder?
Double helix.
So here's the double helix, here's the 2 sides, but the reality is 95% of the time the DNA is not accessible. Okay? Can I transcribe this DNA when it's like this?
No. Nuh-uh.
Can I replicate it when it's like this?
It'll be hard.
Alright? You can't. It's a physical barrier. Now if we add in, I'm just going to grab some kind of prop here. We add this, and we take this protein is something called a histone, and we wrap the DNA around, and then I say I have a long list there. Can you grab me, bring me that one over there, in the chair. Bring that DNA. Alright, so, can you hold this? So you see the little marker, that represents something called a histone, where the DNA is kind of coiled, and then say this wraps around another protein, and then this keeps wrapping and wrapping. So there's proteins that hold it in place. That's really what chromatin is. And then now we further bunch it down when we make a chromosome. So let's see, I'm going to let go of this, throw that out of the way. Alright, so now I'm going to get rid of this. We had it twisted. The first thing you got to do is unravel it. There's an enzyme called helicase that does that. And then there's things called single-banded or single-stranded or single-bonded proteins. Now I have to look that up and double-check that. So let's zoom in. When we say unzip, just for the sake, got a smaller model here. So if this was coiled, helicase unravels it and holds it open. Now I've got access to this. I've got access to this. So now I can make a new DNA molecule. I can do it like this. I can get a new right side. I can get a new left side. Except later on, next week, we're going to call it complementary strand. We're going to call it lead strand and
lagging strand, yeah.
Because the lag strand is the one that actually happens next. The lead strand goes first and continuously. Okay. Good. All right. So as we're doing this, so DNA makes copies of itself. That's replication. All right. DNA is also going to do this. Here's your messenger RNA. It's going to unzip like it did for replication, but it unzips. This time, act like the right side is not there. This time, we're going to call it the coding sequence. It's going to make your messenger RNA. The sugars and the phosphates are going to form. Once the hydrogen bond between the nitrogen bases attaches, and then this sugar phosphate, the phosphodiester bond forms, and this is going to leave. DNA is going to zip back up and coil back up. Now, what I haven't mentioned is there's other enzymes that add the nucleotides. We'll do that next week. They're called polymerase. Right? Remember that term polymer, monomer?
Yeah.
Mm-hmm.
A polymerase is something that adds a polymer, or makes a polymer. Specifically, DNA polymerase. There's an RNA polymerase. Alright, so let's say this. So now we're in the nucleus, which means I better grab this DNA back. The only time this would not be in the nucleus is if it condensed to a chromosome during mitosis, because the nuclear membrane disappears. It's a physical and protective barrier. So I'm going to just hide this down here. The messenger RNA has been made. What's that process called?
Translation?
Messenger RNA has been made.
Oh, transcription.
Transcription. DNA is replicated. DNA is transcribed. So here's my nucleus. You guys get ready. Okay. So now imagine this bubble. I did this earlier in class. So now here's a little hole, a nuclear pore. The messenger RNA comes out of the pore. Now where am I floating around in? I'm in the cytoplasm. Okay. Now here's a ribosome. Hello, ribosome. Okay, so we're going to do this, and we practiced this earlier, so hopefully this will work. Now, this is where it's a little bit of sleight of hand video-wise, but are we in the shot?
Yeah.
Okay. Okay, so here is the messenger RNA, here's the tRNA, and this is what happens. It moves its way down. Okay, it moves its way down, and what happens, so say I'm here, if you can hold it right there. So now, what's going to happen is the next, this messenger RNA is right here. This is the tRNA. There's the amino acid on top. Once it drops it off, this transfer RNA is going to let go. It doesn't have an amino acid in there. What's it going to do? It's going to go pick up amino acid floating around in the cytoplasm. And it's constantly doing that. It can only pair up with messenger RNA through base pairing. So where A is in the RNA, what would the tRNA be? U. Because this is nitrogen-based R to R, or RNA to RNA. Okay, so it still pairs up the same way DNA pairs up with DNA for replication. DNA pairs up with RNA for transcription. Okay, so now see if you can envision this, and the animations are far better. I'm going to rotate this. So as this is happening, as we move down, as this moves down, you're building and building and building and building and building. Let me try that one more time. As it moves down the messenger RNA and tRNA are connecting, and it moves down. Actually, I should say as a ribosome is doing this, this is the final product. There'll be a stop codon, right? This will stop, and then now you have your amino acid. You've gone from DNA to messenger RNA to amino acid. Does that make sense?
Yes.
Okay. Even though I didn't do a good job holding them all, they did a great job. Now, what you can't see is, say my hands were doing this. My hands are the ribosome. So without seeing the messenger RNA and tRNA, hold up a little bit higher. So imagine in there what you can't see. If you were to zoom in, tRNA is coming in, say, here. Okay? So, and then as my hands, as the ribosome, so my hands are the ribosome. As my hands move down, this protein chain is getting longer. Does that make sense? All right, so I just kind of forego using that. Just imagine that being stop codon there. Now I've got a protein that might fold and become a functional protein. Maybe a glycoprotein, or maybe it's a fibrous protein, like in muscles. Of course, if I was using this, I need a lot more fibers to show the guns here. Okay, let's do one last thing. The ribosome is done now. This messenger RNA, because it's the ultimate recycling, it's going to go back to the nucleus. So remember, this is the nucleus. As it goes back in, right, you now have free nucleotides of messenger RNA. So the next set of DNA that needs to be transcribed can use those. It's the messenger RNA is going here, made, produces an amino acid during translation, goes back, gets cut into pieces, get reassembled for the next gene that needs to be transcribed. And that's how we go from DNA to protein. Does that make sense?
Yes.
All right, let's go.